Hello everyone, welcome back to my video. In this series of presentations, the mechanism of translation will be discussed. Translation. We know that this is the synthesis of protein. By transcription process, the informations which are encoded in the genetic material will be transcribed into the mRNA molecule. From the mRNA, these informations has to be decoded into the form of proteins, and these proteins are important for the expression of a character. So this is part of gene expression, or it's otherwise the second and the last stage of the central dogma reactions. So in translation reaction, we need a lot of things. It is common in prokaryotes and eukaryotes. That means whatever be the cell type, all living organisms will be performing translation as a vital mechanism. Without translation, no cell can survive basically, because proteins are the vital part of life for the execution of various reactions, including metabolic activities and other reactions. And as we know that most of the enzymes are even proteins. So what are the requirements for translation? The most and primary requirement is the mRNA itself. Then we need the tRNA molecule to transfer the third requirement, that means the amino acids from the cytoplasm, into the fourth requirement called the ribosome. So the ribosome is the site where the translation event will actually occur. And so we need the mRNA, tRNA, ribosomes, the amino acids. And additionally, we need a number of proteins. Usually the number is very huge. Usually in a eukaryotic or in a prokaryotic system, more than 250 large or macromolecules are associated with translation event alone. And we know that there are large number of tRNA molecules present in the cytoplasm, and also large amount of the large quantity of amino acids are also being stored in the cytoplasm usually. So altogether, majority of the time and materials of a cell is being invested in the protein synthesis. So that is the significance of translation. And moreover, you can also note that much of the energy tapped by a cell will be consumed for the synthesis of proteins. For every small peptides, we need to spend, or the cell need to spend, a considerable amount of energy, right? So when we talk about the mRNA molecule, already we have seen in the previous videos that this mRNA molecule is having a 5' end, a 3' end, with some already modified structures. The introns have been removed. And what is actually inside the mRNA? These are purely the nucleotide base sequences or the nucleotide sequences. So these sequences are the actual informations, right? So these informations are in what form they are decoded. And the answer is that the answer is being cracked during or in the year 1961 by Heinrich Matthaei and Marshall Nirenberg, and they discovered that there are sequences in the mRNA which act as the codon or the genetic code, and these are triplet sequences of the nucleotides in the mRNA molecule. And these triplets, or each of these triplets, will code for a particular amino acid, right? And as we are having 4 nucleotide bases, a total of 64 codons are present with the 3 combinations, right? So out of these 64 codons, there is one start codon, usually that will be the AUG, coding for the methionine, and 3 stop codons are there. And rest of the 61 codons will be coding for a particular amino acid. And please note carefully that we have 20 amino acids, right? We have only 20 amino acids, but we have 61 codons to carry these 20 amino acids from the cytoplasm into the ribosome. So how they are correlated? So it is pretty clear that an amino acid may be coded by more than one codon. Only then the calculation will be okay, right? So there are certain amino acids which are encoded by more than one codons. But carefully note that there is no codon which codes for more than one amino acid, right? So similarly, there are a lot of other features also for the genetic code, right? So in short, so all these different mechanisms, when ready, the cell will start synthesizing a polypeptide using the information stored in the mRNA molecule. And these protein or these polypeptide will be subjected for some processing and will be transported to the corresponding location and where it will perform their function, right? So this is about an introduction to the translation. So shall we move to the next part or the content of translation? Translation as a whole is being split for understanding into 4 stages. First one is the activation of amino acid, then initiation, elongation and termination. So we'll start with the first stage. Activation of amino acid is a 2-step process. The first stage is the amino acylation. That means the amino acid will be charged with an ATP molecule or charged with a high energy compound, right? So the amino acid become amino or acylated amino acid. There is an AMP usually added onto an amino acid. The second stage is the charging of amino acyl AMP to the tRNA. So this is a 2-step process in which an amino acid is first charged with an AMP, then this amino acid AMP or the amino acyl AMP will be charged into a tRNA molecule, right? So here is the mechanism. The first stage is the addition of AMP molecule. You can see the amino acid is being charged by the AMP molecule. They consume an ATP and you will get an amino acyl adenylate or amino acyl AMP. And in the second stage, the charging of the amino acyl AMP to the tRNA, that occurs with the help of a class of enzyme called amino acyl tRNA synthetases. And there are 2 classes of amino acyl tRNA synthetases, class 1 and class 2 enzymes. This class 1 is responsible for the charging of certain set of amino acids given over there, and class 2 is responsible for another set of amino acids. So anyway, whatever be the class, the reaction is almost the same. So in the charging or the addition of this acylated AMP into the or amino acyl AMP into the tRNA, there is a transesterification or there is a nucleophilic attack, and finally you will get an amino acyl tRNA molecule, Right. So the first stage is the addition of AMP to the amino acid. The second stage is the charging of this amino acyl AMP into the tRNA molecule to produce an amino acyl tRNA molecule. Right. And this is an energy consuming process. And there are different types of amino acyl tRNA synthetases. At least 20 different types are present in every cell. And these amino acyl tRNA synthetases are important because these are doing a kind of proof checking. There are two things to be noted. One is that the interaction of tRNA synthetase with the tRNA should be correct and precise. Right. Only then the correct amino acid will be taken for synthesis. The second one is the amino acid should be, or the correct amino acid has to be charged to the tRNA. That's also important. Right. So the two important things in this stage, or the first stage of reaction, is that the proper tRNA has to be in contact with the tRNA synthetase. One. The second one is that the correct amino acid must be incorporated into the tRNA. And because of these two proof checking capacity, sometimes the tRNA synthetases are considered as a second genetic code. Right. You can see the structure of an amino acyl tRNA, a typical structure. Right. And on the tRNA molecule, usually the 3' end of the tRNA is usually involved in this reaction. And we know that at the 3' end there is a CCA residue at the extreme 3' end. And the adenine at the most extreme will be involved in this reaction. And here we can see certain common facts, that the tRNA, we use certain notations. The tRNA with a superscript of amino acid is usually indicating that this is a tRNA which is designed to carry that particular amino acid. Right. But when the name of the amino acid is given also as a prefix of the tRNA, then that indicates this tRNA which is designed to carry an amino acid is now carrying the amino acid at the 3' end. Right. It will be clear here. Here example, tRNA alanine, when it is charged with an alanine, then it is indicated as alanyl tRNA alanine. Right. Hope it's clear. Then in bacteria, so in this presentation we will be focused on bacterial translation. Right. So in bacteria, the first tRNA to be used for translation is tRNA fMet. That means the tRNA responsible for carrying fMet. So what is fMet? fMet is nothing but formyl-methionine. That is the first amino acid to be added for protein synthesis. It's a formylated methionine, not the normal methionine. Right. So these fMet, tRNA fMet will be the first tRNA to be used for protein synthesis in bacterial system, which is not the same in eukaryotes. And also the internal methionines will be taken, or the internal codons for methionine will be identified by different tRNAs, which is denoted as tRNA-met, not fMet. And how this formyl-methionine is formed? It is a two-step process again, or the formation of fMet-tRNA fMet is a two-step process. In the first stage, methionine, the normal amino acid, interact with the tRNA fMet, as we have already seen. That means there is a normal reaction, that there is a charging. you to get methionine tRNA fMet with the consumption of energy. And in the second stage, the methionine which is bound over the tRNA will be formylated with the help of N10 formyl tetrahydrofolate. It's an enzymatic reaction, and that formyl there is a transfer of this formyl group into the methionine to form the formyl methionine. So this is the initial stage of activation of amino acid and charging of tRNA with the activated amino acid.